Okay guys, so let's do some data analysis. At first we will go over with an online tool called as Galaxy. It also has a standalone application, but later on I would expect you to go through these softwares through using your Linux commands, so which we will cover in some later half of this course. Galaxy is an open source framework which integrates many databases and different tools. They are put together in a cohesive environment where you can do different comparative genomic tasks. So here is if you go to Galaxy website, first of all you see this page where we have different options. So you can use Galaxy while clicking this link. You can get its application on your system. You can learn through different tutorials. You can get involved into their projects, so they have a lot of room for developing new tools and putting them together. If we look into Galaxy interface, it is divided into three pans. Left pan is where the softwares and tools are there. In the middle you can see where different menus appear, so you can choose from those menus, and towards right is the history of your ongoing projects. Here is how it looks like. So towards left, if we look towards left hand side, we have these tools which are there. Then we have data display, and obviously you can choose menus from here, and history is where it can give those your different tasks different numbers, and you can view them. You can download your results and reports from here. Galaxy recognizes and interplay among different file formats. You can learn them if you click this link. Different sources of data it can uses. Sequences and base quality data can be stored as FASTQ files and FASTA formats. Sequence alignments can be processed as SAM and BAM formats. Now, whereas genome annotations, we see GTF, GFF and BED formats. So as we go along, when we start dealing with the actual data, we will learn more about these formats. There are other formats which Galaxy can also recognize. What we can do with Galaxy, we can upload our data sets and we can do some initial analysis on it. But obviously we can do that for small data sets. So we can get the data from different databases also, and since they are linked with one another. And as far as data loading is concerned, we can directly upload our data from a local directory, but for big data sets we might want to use transfer protocols, file transfer protocols, FTPs or HTTPs. Here is how we can load our data. If you look into the left pane, we can have Get Data tab, so we can click on that. It leads us into this page, so where we can have upload file option, and we can browse from our local directories. If we click this browse button here, it can also ask us to specify that what kind of genome is it. So it's a mouse or human or whatever is it. So we need to also specify that. Once we press execute, it can upload our data. As far as the file format is concerned, we can specify which file format is coming, or we can put it, or we can leave it as default, which is auto detect, so it can detect which formats are there. We can also provide some address to some URL where it can upload the data from any online resource also. So far what we have seen here is that Galaxy is an open source tool to perform variety of tasks on genome data, and it can accept data in different formats, and data can be directly uploaded via internet also.